Alrighty, hello everybody. Apologies for canceling workshop today. Not feeling so great. Caught some sort of stomach bug, and yeah, I'm gonna record this right now and then immediately post it. So yeah, let's go ahead and get started. I guess real quick, going over the exam from last year for biochem. So if you haven't done it, you like to do it, feel free. I'll explain my answer choices and why I think some answers are wrong, and why the ones that are right are actually right. So yeah, let's get into it. So the way the exam is formatted, at least for last year, was we had like a bunch of multiple choice questions and like one short answer question. So I expect it to be somewhat similar to that in terms of how it's made. So yeah. Starting with question 1, we're saying, which of the following scenarios best demonstrates the flow of genetic information? Okay, so the way I like to tackle multiple choice questions is I like to think about what the answer should be before I even answer. It's good practice for any multiple choice exam, even the MCAT is a good example. So for genetic information and the flow of genetic information, I would say that what you should be looking for is something going from DNA to RNA to protein. That is the best version of genetic information. So let's go through the answers. So DNA sequence encoding a non-coding RNA that regulates gene expression. Okay, that's reasonable. I mean, we're not going from a DNA to an RNA to a protein, but we are going from a DNA to an RNA that regulates gene expression. So that could be something like a non-coding RNA, like maybe a microRNA, something like that. So I can see that being reasonable. Protein folding to assume is no, because this doesn't work because there's no mention of like DNA in here at all, right? So no mention of DNA to RNA to protein or any of that, like DNA to RNA to regulation. So a lipid mediating membrane signal during cell communication, no, not really about genetic information. Carbohydrate polymer being used for energy storage, no, energy storage is not really about that. tRNA molecule binding to a ribosome during translation. I mean, that's a decent answer. I mean, a tRNA molecule, that's, we're going from DNA to RNA to protein. I mean, that's kind of going from RNA to protein. But I think the issue here with this is, this is only looking at one aspect of genetic information flow. It only looks at the translation portion of it. I want something that looks at everything. So, with A, I think A is the best answer here because A is like a DNA sequence encoding a non-coding RNA that regulates gene expression. Specifically, it talks about a DNA sequence that then goes into an RNA, and then that RNA goes and regulates other genes. So I like that answer the best. E is probably not the best because, a tRNA molecule that binds to a ribosome during translation, that only really talks about the translation portion and not the parts before it, unlike A. So I'm going to go with A for that one. How does the presence of a modified base, such as 5-methylated cytosine, affect the structural properties of DNA? So for this one, I'm looking for anything that has to do with messing with the major or minor grooves. Because you guys talked about this, methylation and any kind of editing to the bases in DNA has some sort of impact on enzyme binding and other protein bindings. So anything that has to do with that. So it increases the strength of hydrogen bonding. Okay, well, definitely it has no effect on hydrogen bonding, right? It's not even close to the hydrogen bonds when you're methylating the 5' carbon, so it doesn't matter. Reduces the flexibility of the sugar phosphate backbone. Methylating the cytosine has nothing to do with that, so I'm not going to even entertain that answer. It alters the helical groove, affecting protein recognition. Okay, that's pretty much exactly what I was talking about, so I'm going to circle that for now. This makes sense, right? If you methylate stuff, you're going to change the structure of the major or minor groove, or usually the major groove in this case. And, by changing the structure, maybe some proteins can't bind properly. It prevents DNA from forming non-B conformations. No, that doesn't matter, right? It doesn't matter if it's got a methylation or not. It can still form any other conformation it wants. And it facilitates transition to A-form DNA under physiological conditions. No, I don't think so. I don't really know when that is ever true. I mean, if that's the case, then I would expect all of my DNA to go from B to A, under normal conditions, because there's a lot of DNA methylation that goes on, so that doesn't really make any sense. So I think C is the best answer there. Which sequence would be most likely to increase the melting temperature of a DNA duplex? DNA duplex just means DNA double helix, if you want to think about it like that. I'm just looking for anything that has a very high GC content and is very long. So let's look at A. Bunch of A's and T's. Okay, let's compare A to B. B is a whole bunch of GCs. Okay, so I prefer B over A, so I'm going to keep B. For C, C looks around the same length as B, but there's a bunch of ATs in here as well, so I'd probably keep B as the answer for now. D is the same deal. There's a bunch of A's and T's, so probably not the biggest fan of that. And then E is also the same thing. It's got some T's and A's in there, so I probably wouldn't say that. GCs, remember, have the highest melting point because GC base pairs have 3 hydrogen bonds, while 2 AT base pairs have only 2. So, the more hydrogen bonds you have, the more intermolecular interactions there are between the bases, and therefore the higher the melting point. So I'm looking for anything that has a high GC content. If I'm looking at lowering the melting temperature, I would need more ATs, but I'm looking for the increase in melting temperature here, not the decrease. So, which feature explains the ability of RNA to form more stable tertiary structures than DNA? Okay, so I assume this has to do something with like intramolecular base pairing. So there's cases where RNAs are able to fold on themselves and they can form these kinds of tRNA structures, if you will. tRNAs are actually a tertiary structure of RNA. So they can fold on themselves and make these structures. DNA is exclusively pretty much double-stranded. So it's pretty much never going to be anything else other than a double strand. So I would say that anything to do with RNA kind of forming its own structures with its own intramolecular bonding is like intramolecular hydrogen bonds where it kind of folds on itself and stuff like that. So RNA's ability to form a Z-form helix. Okay, this has nothing to do with explaining the ability of RNA to form more stable tertiary structures. RNA's propensity for intramolecular base pairing and non-canonical interactions. Okay, I like this one. I like this answer. RNA has the ability to fold on itself and do all sorts of weird stuff. It can base pair with itself. DNA, not really. DNA is usually double-stranded. It doesn't fold on itself, stuff like that, without the help of other proteins usually. So, DNA's lack of a 2' hydroxyl group increases rigidity of the DNA helix. That's actually not true. The lack of the 2' hydroxyl group actually makes it more flexible, I would say. I can't remember exactly if it makes it rigid, but it does help with the fact that the DNA won't self-hydrolyze itself. That's one thing to keep in mind. So that also doesn't explain why RNA forms a more stable tertiary structure as well, right? Because that would suggest that DNA maybe actually forms a more stable tertiary structure because it doesn't have a 2' hydroxyl group and it can't hydrolyze itself. So that doesn't explain why RNA has a more stable tertiary structure. So I'll probably get rid of that one. RNA's extended length compared to DNA in the cell. This is just not true. DNA is freaking long. There's some RNAs that are the same size as DNAs. DNA is the same size as RNAs. It's just not true to say that RNAs have extended length compared to DNA. And DNA's rigid B form under cellular conditions. That doesn't really explain why RNA forms more stable tertiary structures. It says that DNA forms rigid B forms, sure, but that doesn't explain why RNA has a more stable tertiary structure. I would need it to say something like, the RNA has a very stable A form because of, extra OH bonding from the 2' OH or something like that. You know what I mean? This doesn't really explain why RNA has a more stable tertiary structure. It kind of just says that DNA has a rigid structure under the cellular conditions. So I'd probably go with B as the answer to that one. What determines the specificity of restriction enzymes for their target sites? For this one, I'm just looking at anything to do with palindromic sequences. Remember, palindromic sequences are when you can read a sequence of DNA the same way backwards as forwards. So the reverse strand is the same as the forward strand. So can't really think of a good example right now, but let's see here. Like maybe G, I think G, G, A, C, C? I think this works. Did I do that right? Oh no, sorry, wait. Let's just do G, G. Like G, 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 C, C, C. Right? And then if this is 5 prime, this is 3 prime, and this is 5 prime for the bottom three, and 3 prime for this side right here. You can see that this is the... So G, 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 C, C, C. So this would be a palindromic sequence, right? So you can read it the same way forward as you can backwards on the reverse strand. And so they would cut some, I don't know, do like a cut somewhere here like this or something, right? Some sort of cut like this. Ah, shoot. Something like that. Okay, so I'm looking for anything that talks about palindromic sequences. What determines the specificity of restriction enzymes for their target sites? The sequence of the minor groove. I mean, that's technically true, except for the fact that it's talking about minor groove. Remember that these things don't bind to the minor groove, they bind to the major groove, so don't like that answer. The methylation pattern of adjacent DNA regions where cytosine is followed by guanine. So, this is talking about like CPG sites. There may be some proteins that are looking at that, but restriction enzymes usually target palindromic sequences. So I'll keep B for now. If there's no better answer, I'll probably go with B, but let's move on to C. Palindromic sequence and hydrogen bonding in the major groove. Okay, I like this. Palindromic sequence and hydrogen bonding in the major groove. Very specific. That's much more specific than B, and also fits what I'm thinking about when I think of restriction enzymes. When I think restriction enzymes, I'm thinking palindromic sequences. I'm thinking something like C. So I'm actually going to say that C is a better answer than B. The density of supercoiling... No. Supercoiling, I mean, sure, probably has some effect on it, but it doesn't determine the specificity of the restriction enzyme. The specificity comes from the actual enzyme itself. The presence of single-stranded DNA regions. No, this has nothing to do with restriction enzymes. You know, restriction enzymes, they look for palindromic sequences. I don't really care about any of the other stuff. It's going to be C as the right answer here. Which step in the ligation mechanism requires ATP hydrolysis? Ah, okay. So this requires a bit of knowledge and, you know, just remembering how the enzyme actually works. So remember that the way that we do this is you basically have for the ligation enzyme, you have your broken strands, right? You have an empty phosphate. And what you end up doing is you actually end up forming this like AMP kind of intermediate with the enzyme where like the enzyme attacks ATP, releases pyrophosphate, and forms a covalent intermediate with the AMP and the enzyme, and then the pyrophosphate is hydrolyzed. So that's the step that requires ATP hydrolysis. And then after that, right, because you've formed this kind of weird phosphoester, the 5' phosphate, I can't remember I think it's the 3' or 5' phosphate. The DNA is able to basically act as a nucleophile to attack the I'm trying to remember the 5' thread. I believe it's the 5' phosphate of one of them, but like the 5' phosphate basically attacks the enzyme, and then the enzyme basically becomes a really big leaving group, and so you can use the OH of the 3' end of the broken DNA to attack the enzyme, kick out the enzyme, and then regenerate the bond. So I'm looking for anything that talks about a covalent AMP thing. So yeah, activation of the 5' phosphate group. So the ATP isn't used to activate the phosphate group. The 5' phosphate group is there, but it's not used to activate it. Formation of a covalent AMP enzyme adduct. Okay, well, I don't really know what an adduct is. I'm going to make the assumption that it means, like, combination. You know, even if there's words that I don't know, I can make an assumption based off how the answer is phrased. So I like that one a bit better. Base pairing of the DNA ends to be joined. What the heck? Which step requires ATP hydrolysis? No, the base pairing part does not require that. Annealing of the complementary strands. So, okay, complementary strands important here. I don't care about the complementary strand when I'm doing the ligation. The ligation only ligates one strand, remember. You only break one strand here. Dissociation of the ligase from the prepared DNA. No, the ATP is not involved in dissociation. It's probably going to be B in that case, right? Formation of the covalent AMP enzyme adduct. That makes the most sense there. So, yeah. For question 7, we are looking at linking number. Okay. So in this case, we're going to look at linking number. I'm just trying to find the image here of the enzyme, the ligase enzyme, actually, so I could show you guys what I'm talking about. But I don't know. Do I have it? Oh yeah, I think I do. Here, wait. Let me try and show this to you guys real quick. Where is the ligase? Here it is. I don't know if you guys can see that. Can you guys see that? Oh, you can't. Hold on. Switch this real quick. Nope. Not that. No. Here. Maybe if I switch this and show this to you guys. There we go. Okay, cool. Nice. All right. So here's that enzyme thing I'm talking about. So look at this, right? Remember the ATP comes in, forms this covalent AMP adduct, and then what happens is the phosphate, right? This phosphate acts as a nucleophile, right? And attacks the other phosphate right after this AMP adduct, and then it basically the enzyme becomes a massive leaving group. Right, so that's what I was talking about. This is what the ATP is used for. The ATP is used right here to make this AMP covalent enzyme thing. So that's what I was talking about there. All right, back to this. So, a relaxer has helical 500 helical turns. Okay, cool. Introduce 50 negative supercoils. What is the linking number? Okay, so remember that the linking number is equal to the number of its twist plus writhe equals linking number. And remember that DNA is, usually coiled in the right-hand direction in terms of twist. So twist is right-handed, and generally speaking, when I'm doing twist plus writhe, the writhe is usually right-handed as well. So if I have 500 twist and some positive writhe, right, it's going to be 500 plus 50 is going to be 550 linking number. But this is different. This is 500 helical turns, 500, and 50 negative supercoils. Negative, right? It's not positive, it's negative. So it's actually going to be minus 50 equals my linking number, which is just going to be 450 in that case, right? 500 minus 50. Remember, if this was positive supercoils, then it would actually be plus. It would be 550. But because it's negative supercoils, it's going to be 500 minus 50 in that case. So it's going to be B. Why are chain terminators such as dd-NTPs effective in DNA sequencing? Okay, I'm trying to find an answer here that has to do with the fact that they they basically, in Sanger sequencing, what they do is that you have a dye on them, right, and it stops the elongation of a chain. And when you stop the elongation of that chain, you can then use it in a gel electrophoresis to kind of figure out what kind of base is at each position. So that's why the dd-NTPs are effective in the DNA sequencing, is because they basically allow for you to check what each fragment is made of and what base is at each position. So, yeah. They prevent DNA polymerase from... No. They don't prevent DNA polymerase from binding to the template strand, okay? The DNA polymerase can still bind, right? Nothing changes about the binding. They lack 3' hydroxyl group, preventing elongation of the synthesized DNA strand. This is true. They do that. They do prevent the elongation of the synthesized DNA strand. And that is actually how Sanger sequencing works, right? Because you add a dye to these dd-NTPs, what ends up happening is you just add these dideoxys, and when you've added these dideoxys, you completely terminate that chain, and then you basically do that over and over again until you get to the next. You just keep doing it over and over again, and then when you put it all into a gel, you'll get all sorts of different lengths of DNA fragments that are different colors, and the different colors represent which base is at each position. So, yeah, I like B. They introduce mutations. No, they don't. They don't introduce mutations, right? Nothing changes about the bases, right? The bases are still the same, and the complementation is still the same, so that doesn't change. They cause DNA polymerase to dissociate. No, they don't. They don't cause any. They don't affect DNA polymerase's ability to work, right? I guess like after it has been bound and like is attached to the DNA, sure. I guess it stops DNA polymerase from continuing to polymerize, but it doesn't stop It doesn't stop from dissociating. They stabilize mismatched base pairs in the active site. No, doesn't make any sense. They don't stabilize mismatched base pairs. I mean, mismatched base pairs is a completely separate topic. It's not really involved with the whole DDNTP process. When you see DDNTPs, you should be thinking of Sanger sequencing. If you see reversible chain terminators, you should be thinking of Illumina sequencing or next-gen sequencing. So I'd say B is the best answer here. How does the geometry of DNA polymerase active site promote replication accuracy? Okay, so, I mean, I guess I'm looking for anything here to do with, like, the exonuclease activity or anything to do with preventing like normal nucleotides from going into the DNA polymerase instead of DNTPs. You don't want normal nucleotides to go in there. You want DNTPs to go in there. So anything to do with the exonuclease activity, and anything to do with only having being very specific for the 2' deoxy form of the ribose sugars. Okay, so by discriminating between ribonucleotides and deoxyribonucleotides. That's pretty true. That does explain the geometry. Right, remember the geometry of DNA polymerase, very specific, so that it doesn't pick up any ribonucleotides that have the 2' OH group. So I like that answer. It's a good answer. It does discriminate between ribonucleotides and deoxyribonucleotides. By stabilizing mismatched base pairs through hydrogen bonding. That actually wouldn't promote replication accuracy. That actually kind of goes against that. If it stabilized mismatched base pairs, that doesn't actually go with replication accuracy. That actually kind of decreases replication accuracy. By increasing rate of replication to minimize errors. That doesn't really explain how the geometry of DNA polymerase promotes replication accuracy, right? I want an answer that talks about the geometry, right? That's not really a good answer compared to A. By allowing non-Watson-Crick base pairs under certain conditions. Again, no. That would suggest that it has a decrease in accuracy. I want more accuracy. I don't want this thing to increase or the increase in errors, right? I want it to be a decrease in errors. By cleaving pyrophosphatase to release energy for proofreading. It's a decent answer, but it's not talking about geometry. Again, I guess it's true. It does cleave pyrophosphate to release energy for proofreading, but it doesn't really talk about the geometry. So I would need something about geometry. And does this discrimination between the ribonucleotides and deoxyribonucleotides, that's a very specific geometry for the enzyme active site of DNA polymerase. So I would say that's the best answer here. Which of the following enzymes is specific to chromosomal DNA replication, not involved in the process in other processes, and absent in PCR? Okay. So let's think about this here. Which of the following enzymes is specific to chromosome? I'm trying to remember if you guys have actually done this. It's specific to chromosome replication, not involved in other processes. Okay, I don't know. I don't think you guys have actually done DNA replication yet. I'll go over this question anyways. But yeah, so let's take a look at this, right? So, specific to chromosomal replication, not involved in other processes, and absent in PCR. Okay, so I think I actually got this question wrong on the exam. But yeah, let's take a look at this. So for looking at like DNA polymerase, obviously DNA polymerase is not specific to chromosomal replication. It's used, not involved in any other processes and absent in PCR. It is present in PCR, right? So you can't use that there. But yeah. I think the main thing that I'm thinking about in PCR, so what does PCR use? PCR uses, it needs primers, right? And primers are like these, like, in this case, it uses DNA primers, remember. And the DNA primers are, complementary sequences to the DNA that you use to anneal to your DNA and then elongate your DNA. So anything that makes those primers is not going to be useful in our PCR setup, because you remember in normal DNA replication you actually have primase, and that's actually one of the options here. B is primase. Primase is actually used in normal replication of the DNA, and you need that because what it does is it actually lays down these RNA primers, right? Those RNA primers are what's used to kickstart chromosomal replication, but that's not really the case in this here. So, yeah. The other thing I should point out is helicase. Helicase is also used in replication. This is true. So it is specific. But it's not specific to chromosomal replication. It's actually also used in transcription. It's actually used in transcription because, and remember, in transcription, you also have to split the DNA so that the RNA polymerase can get in there and actually do the transcription. So helicase is actually not a good answer here. Sorry, I just have to look at something else. Okay. Anyways, for ligase, so specific to chromosomal DNA replication. Okay, ligase is something that is used in DNA replication, but it's not specific to DNA replication. It's also involved in other processes, and by that I mean it's involved in just general DNA repair, right? So any kind of repair that you're doing, you can use ligase for that. So ligase is not a good answer here because ligase is just a general repair enzyme, and it's not very specific to DNA replication. It's used in many other processes that just involve any kind of repair. Exonucleases is a bit of a weird one, but it's one of those things again that you need to remember it's not very specific to DNA replication. So exonucleases are used in all sorts of things: DNA repair, RNA degradation, like breaking down RNAs. But especially in terms of DNA repair, they're very common. So they're not specific to DNA replication, even though they are used in DNA replication, right? Just to make sure that we keep our fidelity and our accuracy. But they are definitely involved in other processes, so that doesn't satisfy the second thing here. So I would say exonuclease is not a great answer. So that leaves primase as our best answer here, and primase works the best here because remember, it's not involved in any other process. You never have to use an RNA primer for anything else, and it is absent in PCR because in PCR we make our own DNA primers. So there's no need to use an enzyme that makes primers for us. Okay, which nitrogen atom position is suitable for modification with fluorescent dye in the fluorescent chain terminators? Oh, okay. The freaking wording of this really threw me off for a second. I was like, what is this talking about? But yeah, I mean, this is kind of one of those things that you just have to know. If you try and put a fluorescent dye at A, E, D, and C, it'll actually mess with the structure significantly. Because remember, these, especially these guys here, E, D, and A, these are heavily involved in like actual H bonding. So if you try and put a big dye in there, it's actually going to screw up the structure, and it's going to mess with all the interactions that make this a very stable molecule of DNA or RNA, right? You're going to mess with base stacking, you're going to mess with hydrogen bonding. It's just not going to be a fun time. Same thing with C, especially for base stacking. C is not very super involved in hydrogen bonding, but if you put the dye on C, you're probably going to have issues with base stacking, because remember, these dyes are actually much larger compared to the bases themselves. So, yeah. The best option here is going to be B, because B is like very far removed from the other, from like where the interactions are occurring for the bases, and it's also not going to get in the way of the base stacking. If anything, B is actually going to be, you know, where the major and minor groove are, so maybe it'll affect like enzyme binding, but not only is it maybe going to affect enzyme binding, but it'll actually make it very easy to see the fluorescence, because it's going to be on like the outside of the DNA instead of stuck in the inside somewhere here, if that makes sense. So I'd probably say that B is the best answer here. Okay, number 12. You ran a purified plasmid DNA sample, okay, so plasmid, right, I'm thinking bacteria, on an agarose gel and observed 3 distinct bands, okay, at different positions in the gel, okay. The bands correspond to different forms of the plasmid, base, whatever, yeah. Identify the likely forms. Okay, so this is talking about like supercoiled DNA, nicked DNA, stuff like that. So remember that, you know, the farther down it travels, the more compact or smaller the DNA is. So I'm expecting that the farther down it goes, the more supercoiled it's going to be. So yeah, anything that's like that, I'm going to expect that. And then remember that anything that is like any nicked DNA, right, DNA that's broken, right, is going to be very like rigid, or not rigid, but it's going to be very slow moving, because broken DNA doesn't like move as one unit, if you will. And then, you know, linear strands move faster than nicked strands. Nicked strands are very slow, so I would expect something to do with like maybe nicked first, then linear, and then supercoiled. So yeah, let's start from E this time actually. Unbound nucleotides, circular DNA, and supercoiled DNA. Okay, so first of all, there's no unbound nucleotides. So I'm never expecting to see unbound nucleotides in my gel, because they're too small to show up in a gel, first of all. Now, I need larger molecules to show up in a gel, so it's not going to be E. I don't know why I circled it. 3 fragments of linear represent only fragments of linearized plasmid. No. I mean, linearized plasmid, I mean, maybe, but, I mean, that could be a reasonable answer. It's better than E, that's for sure. I guess we'll look at C first, though. The 3 bands represent linear DNA, RNA fragments, and genomic DNA contamination. Okay, so RNA fragments, probably not. RNA degrades very quickly, usually. So I'm not expecting RNA fragments to show on here. Also, it says purified plasmid DNA samples. Why would I ever see RNA fragments in my plasmid DNA sample? So, no. 3 bands represent single-stranded DNA, double-stranded RNA. Again, RNA, you're not going to see RNA on this. You're running plasmid DNA samples. So no. So now I'm looking at this. The 3 bands represent nicked DNA, nicked circular DNA, linear DNA, and supercoiled DNA. Okay. So, I mean, this is probably better than D. So I would probably say that's the best answer, but let me double check something real quick. Yeah. Okay, so yeah, I'd say A is the better answer here. I had to double check something real quick, the linear DNA versus the nicked DNA. So even though it's circular, the nicked DNA actually takes up a lot more volume and space, which is interesting to think about. But yeah, so actually, funny enough, in this case, I'd say A is the better answer than D, because it doesn't say really anything about, you know, you running restriction enzymes on this. Like, usually they specify that. So I don't expect there to be any kind of restriction enzyme that talks about fragments of linearized plasmid cut by a restriction enzyme. I feel like they would have had to say that in the problem somewhere, that you ran a restriction digest on it. But because there's no restriction enzymes here, I don't think D is the best answer here. So I'm going to go with A instead. I believe, yeah, okay, so these questions here, so 13, 14, and 15 are prime examples of like these multiple choice questions that require a bit of data analysis and understanding of how data works in a lab setting, basically. So we're going to go over these. I'm going to try and explain these. Hopefully they make sense. So 13, micrococcal nuclease, MNase, preferentially digests DNA in regions not protected by proteins. Not protected by proteins. Okay, so it digests DNA not protected by proteins. The only protein I know that protects DNA is histone proteins. In histones, the DNA wraps around them, right? And it protects 147 base pairs, plus or minus maybe 20 or 30 or whatever, right? So we're protecting 147 base pairs from being broken down by the MNase. So, such as nucleosomes. Look at that. When DNA is extracted after MNase treatment and sequenced, the protected DNA fragments, 147 base pairs, correspond to nucleosome-bound regions. Okay. You perform the MNase thing, right? Okay, cool. And they've revealed the following results. Promoter regions show a depletion of nucleosome signal. So there's no nucleosomes around these signals. So I'm going to write that down. So no nucleosome around promoters. It's good to know. Okay. Gene bodies exhibit regularly spaced nucleosome signals. Okay, so that means that genes generally have nucleosome. Or gene bodies means like protein coding regions, basically. So the protein coding regions generally have... the protein coding regions generally seem to have the nucleosomes around them. Strong periodic pattern observed around transcription factor binding sites. Okay. So there's some sort of pattern around binding sites for transcription factors. So it seems like the transcription factors are probably able to bind at least somewhat. So yeah, so it seems like it's digesting the DNA around transcription factor and binding sites, which means that it's not being covered by nucleosomes. TF binding not covered by nucleosomes. Okay, so what this is telling me, right, and the way I got that, by the way, is that, it specifically says that it digests DNA in regions not protected by proteins. So a strong periodic pattern is observed surrounding transcription factor binding sites. Tells me that it basically tells me that there's a lot of cutting going around these transcription factor binding sites, which means that they're probably more exposed than like these gene bodies, for example, because these gene bodies are regularly spaced with nucleosome signals. So I expect that these are a little bit more free and able to be cut by the MNase. So what I'm getting from this data is that it seems like the gene body itself is actually very protected by the nucleosomes, while the actual promoter region and the transcription factor binding sites are not super protected, which tells me that like maybe the initiation of the transcription of these genes is actually a little bit easier, because they're not as tightly like regulated or I guess hidden by the histones, if you will. So nucleosome positioning in promoter regions is disrupted to enable transcription factor binding and gene activation, while coding regions maintain nucleosome arrays to regulate elongation. Okay. I do like this answer. I like the first half especially. Their position in their positioning in is disrupted. So there's no nucleosomes in promoter regions to enable transcription factor binding and gene activation. That kind of goes hand in hand with what we talked about in the first observation and the third observation. So I like that answer. The observed, the M&A experiment indicates that nucleosomes are assembled more tightly around promoter regions. Nope, we just debunked this, right? So promoter shows a depletion of the signal, so there's no nucleosomes around them. It's not B. The observed periodicity around the transcription factor binding site suggests random nucleosome assembly. Doesn't seem very random to me. It seems like it's very specific, where it's like the promoter regions are very specifically not bound by nucleosomes, and there's very clearly a strong pattern where the transcription factor binding sites are relatively open. So I would not call it random. I don't like this word random. So I'm going to get rid of that one. I don't like that one. Promoters have intrinsically high GC content. Okay. First of all, where on earth did it talk about GC content or AT content anywhere in the question? This is out of scope. It's not discussed here, so I would say D is not the right answer. And then MNase, digestion only detects nucleosome-free regions and is unable to reveal nucleosome positioning. Well, this is just not true. If you can digest regions that are free of nucleosomes, then the areas that you didn't digest are going to be the ones that have the nucleosomes. So this is just not true. So in that case, A is going to be our best answer here, and also follows the data very well. So A is the right answer here. For question 14, let me get some water real quick. All right. Question 14. You're studying a linear DNA fragment that is 10 kilobase pairs, okay? And it contains recognition sites for 2 restriction enzymes, EcoR1 and HIND3. You digest it separately with each enzyme, then perform a double digestion with both enzymes together. Okay. The digested fragments are analyzed on an agarose gel, and the following are observed. So, EcoR1 yields 2 fragments. Okay. So, I'm just going to draw some lines here to try and remember each one. So, EcoR1 and HIND3 is 6 and 4. Okay. So, and then a double digestion is that. So, let's do this. Let's do both. Let's do, this is EcoR1. This is HIND3. It should be R. It is EcoR1, but I'm going to put the I there because it's the right Roman numeral. And both. Okay, so let's take a look at this. When we do our digestion, we should look at the first one. EcoR1 digested into 2 fragments, 7 and 3. Okay, let's just draw a line. So let's put it there. This side's going to be 7. And this side's going to be 3. Cool. HIND3 is going to be 6 and 4. Okay. It's like in the middle somewhere here, maybe. 6 and 4. Alright. Double digestion yields 3 fragments. A 4, a 3, and a 3. Okay. That's really interesting, isn't it? So, a 4, a 3, and a 3 is interesting because what that suggests is that this 7, right, is being broken up into a 4 and a 3. Right? So basically what's going on is I know that EcoR1, let's draw it here actually. So we got a 3, 3, and a 4. Oh god. 3, 3, 4. Okay. So I know that EcoR1 cuts here, basically, right? So I'm actually going to keep that in mind. I'm actually going to move that here. So this right here is going to be EcoR1. And I know that because it cuts... It's gonna make a 7 and a 3. So I know, like, if I add these two together, the 3 and the 4 here, it's a 7, and this is a 3 here. So it's gonna cut where this is. Now the only thing is this right here, this extra line here. But if we look here, if I didn't have EcoR1, right, I would have a 3 and a 3, which is a 6, and then a 4, which is what HIND is doing, right? HIND3 makes a 6 and a 4. So I actually think that HIND is going to cut here. right? And if I put both together, what's going to happen is that eco R1, if I put eco R1 in first, for example, it's going to make the 3 and the 7, and then when I put hind 3 in, it's going to make another 3 and a 4. So I'm expecting this to be what I'm looking for in the answer, because the question is asking which of the following maps the restriction enzyme the restriction sites along the DNA. So I expect so I don't expect them to cut at the same position. So like, yeah. So A, for example, says with both enzymes cutting at the same position. I don't think that's the best answer because they don't cut the same position, right? Eco R1 cuts here, hind 3 cuts here. So don't like that answer. Eco R1 cuts 3 bases from one end, while hind 3 cuts at 4 bases and 6 bases from the same end. So they don't cut. So hind 3 doesn't cut twice. None of them cut twice. They both cut once. So I don't like this answer either. Eco R1 cuts 3 bases and 7 bases from one end, and hind 3 cuts at 4 bases, creating an overlapping fragment. I don't like this one either. It doesn't cut twice, just once. Eco R1 cuts 3 bases from one end. Okay, that's true. While hind 3 cuts 6 bases from the same end. That's true. Hind 3 cuts 6 bases from the same end. So if eco R1 cuts 3 bases from this end, which is true, right? 3 bases from that end. Hind 3 is 6 bases from the same end. Right? There's no overlapping sites. That's true. There is no overlapping sites that I'm seeing here. So I like D the best right now. Eco R1 cuts at 3 bases and 7 bases. Again, I don't like this double cutting nonsense, so I'm going to get rid of that one. So I'm not even going to read the rest of it. If you want to, while hind 3 cuts at 4 and 8, creating 3 distinct fragments upon double digestion. They don't cut twice from what I've done here. So I'm going to go with D as the right answer here. For 15, you are tasked with sequencing a 500 base pair fragment using Sanger sequencing method. To ensure proper termination of DNA synthesis at each nucleotide position, you prepare 4 reaction mixtures. Okay, so we've got some sort of Sanger sequencing thing here. We've got a DNA template. Okay, good. We've got a polymerase, a mix of DNTPs, and then a small controlled concentration of DDNTPs. Okay, so far, nothing strange. This is very reasonable. After running the reactions through DNA electrophoresis, you observe that peaks corresponding to T are underrepresented. Okay, so maybe you didn't add enough thymines, like maybe you didn't add enough of the DDNTPs or the DDTTPs. Not enough. So it seems like you maybe didn't add enough of them. The longest fragment terminates at 300, even though the DNA template is 500. Okay, so maybe you either didn't add enough NTPs, or you added too many other DDNTPs. Not enough NTPs, or not enough DDNTPs. Or too many, sorry. Too much DDNTPs. The fact that it's terminating early suggests that you either don't have enough NTPs, or you've added too much DDNTPs and you didn't add a lot of enough TTPs, right? Adding more DDTTP to the reaction increases the number of T peaks, but does not extend beyond 300. Still, okay, so the fact that it doesn't extend beyond 300 is still really weird. So even if you add more DDTTPs, it doesn't change the fact that you're not going beyond 300. That, to me, tells me that we actually have way too much DDNTPs. Too much DDNTPs. And I say that because if we're adding, so the first problem is the fact they're underrepresented. Okay, that probably means that we're out of the DDTTPs and there's not enough of them. Okay, sure, we should be able to just add more DDTTPs and we should be able to continue, I suppose. Second point says that it seems like it terminates at 300 and nothing longer. That suggests that, maybe the ratio is a bit weird. Maybe we have too much DDTTPs, because that would mean that we're terminating a lot, right? If we're terminating a lot, it would make sense that we would never get to 500. Because, we've added too many of the terminators, and the terminators just keep binding at around 300. And adding more DDTTPs to the reaction, it does increase the number of T peaks, which is great, which means that we've solved that problem. But it still means that but the fact that we are still not extending beyond 300 means that it's most definitely an issue with the amount of terminators we have. So I'm looking for an answer that talks about the DD-TTP concentration being too high, or the ratio of DD-TTPs to DNTPs being too high. So concentration of DD-TTP is too low, leading to inefficient termination at thymine residues. I mean, that could be it, but like, I mean, in 3 we're adding the DD-TTPs here, and it seems like there's still a problem of termination. So I don't think that's the issue here, right? Because if the issue was that we didn't have enough DD-TTPs and we were terminating early, then surely adding the TTPs in there would solve that problem. But that's not the case here. We still have the issue, so it's not that. The DNA polymerase lacks the ability to extend past secondary structures in the template, causing premature termination. Nothing here is mentioned about secondary structures in the template, so I don't like that answer. I'm going to skip that. I don't think that's a good answer here. An incorrect ratio of DNTPs to DDNTPs in the reaction is causing an imbalance, prematurely terminating the synthesis. I like this answer. This is exactly what we talked about, right? We said that initially we didn't have enough of the TTPs, the DDTTPs. Second point was we don't have enough NTPs, or we have too much DDNTPs. And the third point was we have too much DDNTPs. This is exactly what this is talking about. Incorrect ratio of DNTPs to DDNTPs. And it causes an imbalance because we have too much DDNTPs, causing termination. So I like that option. I like C. The primary annealing step failed at the 3' end, leading to incomplete extension beyond 300. I mean, nothing tells me that it should have failed, especially considering the data. The data doesn't talk about anything about failing, right? Because clearly it seems like the primers are annealing, so I think it's perfectly fine. The chain terminator DD-TTP is inhibiting synthesis past 300 due to overloading the reaction with DD-NTPs. I mean, I like the second half of this answer, but I don't like this. This is suggesting that specifically the DD-TTP is causing the problem, but I don't think that's the problem. The problem is that just generally speaking, we have too much DD-TTPs. So because of that, I would say that C is the best answer here. Just because of the fact that, you know, in E, it's not that DD-TTP is the problem here, because we added DD-TTPs back in, and the reaction was able to progress, but it still stopped at 300. So the DD-TTP is not the problem here. So I'd probably go with C as the best answer here. All right. 16 is a fill in the blank. You guys had something like this on your Hurley exam. I think it was your third test, I think. But you guys had a word bank. You guys don't get word banks here, so you kind of just have to pray. I would say the best thing to do for something like this kind of question is you need to find a table, and I actually brought this up in one of my workshops, I believe. You need to find a table that has like all of the values for DNAs and the different types of DNA, and you need to copy those things down onto your note card. Because there are some questions here where they just straight up ask for you to recite a specific number. So remember, just make sure that you're doing that. So a DNA polymer is a polymer of repeating units called nucleotides. This should make sense. Hopefully you don't have to write that down on your note card, right? But it's just made by a bunch of nucleotides. They're connected by phosphodiester bonds. Remember, it's a phosphodiester bond. There's like a phosphoester there. There's a phosphate connecting the 3' OH of one side and the 5' OH of the other side, of the other nucleotide, forming sugar phosphate backbone. The double helix is stabilized by blank bonds between bases and blank interactions. Okay, so between bases, I'm looking for hydrogen bonds, and specifically the word interactions. The only other thing I can think about that stabilizes the double helix is the van der Waals interactions or the hydrophobic interactions. So I would probably specifically here say, I'd specifically say like van der Waals interactions. I think you could probably say like base stacking. Base stacking is probably a good answer as well. but I mean, either one of those is okay. I wouldn't say hydrophobic interactions because hydrophobic interactions is a little bit too vague. I know there's only one type of hydrophobic interaction really in DNA, but like we want to be specific here. You could say pi stacking too, right? Because like the pi stacking also works, but you specifically want to say I think either van der Waals base stacking or pi stacking because hydrophobic interactions is a little bit too vague. In the B form, which occur between the stacked base pairs, and that further proves that we're looking for the van der Waals stuff. In B form DNA, the helical twist is approximately, this you just have to know, it's about 3.4 angstroms. Write this down on your note card. I would look at, oh wait, sorry, wait, I just realized, I think I messed this up, wait, sorry, no. The helical twist is 36 degrees. The rise is 3.4 angstroms. So these are just things you have to write on your note card. You just got to write them down. There's no point in trying to memorize these. You could if you wanted to, but considering you guys get a note card, use it. So yeah, I would go with that. And make sure when you do these, you write these down for the A, B, and Z forms of DNA. Write it down for all three of them, not just like one or two. All three of them. Right? In cellular contexts, DNA undergoes... Undergoes? What the heck? In cellular context, DNA undergoes what supercoiling to accommodate the spatial constraints of the nucleus? Okay, so it usually undergoes negative supercoiling, right? Because positive supercoiling increases strain. Negative supercoiling reduces strains and actually makes it more compact. Yeah, the supercoiling is regulated by topoisomerase. That's the enzyme that introduces the supercoils, right? Which can either introduce or relax supercoils. DNA compaction begins with its association with histone proteins to form nucleosomes, right? Nucleosomes, remember, those are the complex of the 8 histone proteins, the octamer plus the DNA wrapped around it, where approximately 147 base pairs are wrapped around an octamer of histones. I tried to hammer this home. It's 147 base pairs. Also something to write down on your note card. If you've done it enough, though, you'll probably just remember it, so I wouldn't be too worried about it. But, yeah. Okay, 17. Choose one of the two phosphoester bonds indicated, but, structure in the following reactions. Okay, so we're basically trying to figure out which bond is being cleaved. So the bond blank is formed by DNA polymerases. Okay, so and remember, in this case, what's going to happen is DNA polymerase is going to use the 3' OH, so this OH here, of one base, and attach it to the 5' phosphate group of the other base below it. So it's going to form bond 1, right? Because bond 1 is the one that's going to be formed after you attack, right? So that OH, right, or this O, I'm going to make it O minus just to make it a bit easier on myself. But that O minus, whatever, right, is going to attack here. I drew it very wrong, but like here, basically, right? So it's going to be bond 1. For the second one, the bond blank is formed by DNA ligase. So DNA ligase is also going to be bond 1. Remember, what you're doing is you're taking the 3' OH of one of the DNA and attaching it to the 5' phosphate of another base. It's going to be bond 1 again. The bond blank is cleaved by restriction enzymes. Again, it's the same thing, right? The restriction enzyme just cleaves this bond here. The bond blank is cleaved by RNA hydrolysis. Okay, this one, remember, self-hydrolysis is when this O comes here, attacks here, and kicks off this bond. So bond 2 is the one that's cleaved by RNA hydrolysis. Because remember what you end up with, I'm not going to draw the whole thing, but you end up with this thing. I drew this horribly. you end up with this thing, right? As the phosphate, right? Because you've broken off the second bond here. So 2 is cleaved. The bond blank is cleaved by topoisomerases. Topoisomerases, they also are similar to restriction enzymes. They cleave bond 1. Right? Most enzyme mechanisms do things by cleaving this bond. Right? The only time that I've seen that's not true is for self-hydrolysis through RNA. That's the only time I've seen it cleave bond 2. Every other enzyme, though, pretty much cleaves bond 1. So, yeah. Okay. For the following question, write your response in the space provided. 18. You isolated a circular genomic DNA molecule. I would honestly, for your guys' exam, I would expect a question like this. It's like the only way they can ask a short answer question, I think, really. Maybe I'm missing something, but like this is one of the easiest ways to ask. So, yeah. You isolate a circular genomic DNA molecule from virus that infects E. coli. You subjugated? You subjected the DNA to acidic hydrolysis to break its backbone and analyzed the resulting base composition using paper chromatography. Okay, cool. The results are as shown. 22% A, 28% G, 36% C, 14% T. Okay. Automatically, I'm thinking, okay, the ratios of A to T is not the same, and the ratio of G to C is not the same. So I am immediately thinking that this is single-stranded, or it's going to be it's going to be single-stranded, right? It's also going to be DNA, because I've got T and no U. So I'm thinking it's going to be single-stranded DNA, So that's what I'm thinking right now. Based on this, what can you infer about the structure or characteristics of the viral genome? Okay, well, the other thing I can also infer from it is that don't really have a lot of AT content, right? There's not a lot of A's, not a lot of T's, but there is a lot of G's and C's. So I'm going to say it's going to be single-stranded DNA with high GC content. Is probably what I would say. Right? So the two things you want to make observations about when you're doing these kinds of questions is the GC content versus the AT content, and whether or not it's single-stranded DNA, RNA, whatever, right? Double-stranded, whatever. That's what I would look at. Provide a detailed explanation. So for this one, I would just say like single-stranded. I'm single-stranded because according to Chargaff's rule, if it was double-stranded, A has to be equal to T and C equals T G. Sorry, but that's not the case here. Something like that, right? Sorry, Chargaff. Capitalize your name. Okay. You design primers to amplify a viral gene using PCR with annealing temperature of 60. However, no amplification product was observed. What could be a possible reason for the failed reaction? So, I mean, if the fact that, annealing didn't work, I think the best thing to say here is, like, maybe the annealing temperature was too low, right? So if the annealing temperature is too low, right, you just won't get any annealing at all, actually. Sometimes, like, it just... Because when you want... For the annealing, it requires, like, a very specific level of heat or temperature. And if it's too high or too low, that annealing is a problem. It's going to be a problem because it's going to anneal non-specifically, right? So it's going to anneal non-specifically. It's not going to be specific to where it's annealing, so it's not going to produce the right product. So I would say it's either going to be too high or too low. So annealing temperature is too high or low. I'm inclined to say too low here because 60 is actually a pretty reasonable annealing temperature, right? Remember, you're generally for denaturation it's about 90. For annealing, it's about 60, 70-ish, and then for elongation, it's about 70, 75-ish. So this is a reasonable annealing temperature. So the fact that it's not working usually tells me it's probably too low and not too high. So I'm actually going to specify that. Which means we're getting non-specific primer binding and no DNA elongation and no PCR product, something like that, right? How could you address this issue to optimize PCR? This is pretty easy. Increase the annealing temperature. Something like that, right? And maybe you could explain, this would allow for more specific binding of the PCR primer. Something like that. You successfully amplified the entire viral genome using optimized PCR conditions. Okay? They then repeated the acidic hydrolysis and paper chromatography to analyze the base composition of the amplified DNA. Based on your characteristic PCR amplification, predict the base composition. Okay, so they have managed to do this PCR, right? And they have made a copy or a complementary strand for this. Okay? So, what I like to do with these and the way I like to imagine this is, let's say that you have 100 bases, right? Right, so 22 are going to be A, 28 are going to be G, 36 C's, and 14 T's. Okay. So remember, these all pair up, and these all pair up, and these pair up. The way that you should think about these is all I have to do is add them together, right? So if I have 22 A's, then I'm going to have 22 T's, right? So I'm actually going to put them all in a line again. Let's say this is my strand, right? My strand is just 22 A's in a row, 14 T's in a row, 28 G's in a row, and 36 C's in a row. That's obviously not what the actual strand is, but for the sake of simplicity, we'll go with that. So for the 22 A's, I need to have 22 T's. For the 14 T's, I need 14 A's. I didn't put any spaces between some of these. It's bugging me. For the 28 G's, I need 28 C's. And for the 36 C's, I need 36 G's. Okay, fair enough. Now all I have to do is just add these together. So let's add the A's and T's together. So the T, so this A has to go over here, the T here, this C, and then this G. Right? So now if I just add these together, this I believe is 36, 36A. Just gonna do this and add a plus sign here to make it a bit more formal. This is gonna be 36 T's. See how these are starting to match up now? Like, look at how the A's and T's are now the same number because it's double-stranded now. 28 and 36, that's 64, I think, if I've done that right. 64 G's. And 64 C's. Great. Okay. So, now all I have to do is do a little bit of math. And I don't remember if you guys get a calculator. I don't think you get a calculator on the exam, unfortunately. But right now, the only issue is that the answers here are asking for percentages, right? And right now, if I add all these together, that's 200. So the easy thing to do is to just divide this by 2 to make it 100. And so if I'm dividing this by 2, I just have to divide all of these by 2 to get my percentage values. So 36 divided by 2 is going to be 18% A. And then if it's 18% A, it also has to be 18% T. For G, 64 divided by 2 is 32%. And then this is also going to be 32%. And that is going to be the answer here. So, and then for the explanation, provide your explanation. You could basically say, you know, PCR generated dsDNA from the original ssDNA given, which means that the newly synthesized DNA strand will allow the A and T's to be equal while the G's and C's will be equal. Done. That's what I would do. So that's the end of the exam, actually. So I hope that makes sense, guys. I tried to go through this quickly, but also try and explain my answer choices for each thing. So I hope it makes sense. But yeah, other than that, good luck tomorrow, because I know your exam's tomorrow morning. And yeah, happy studying.